Good morning, everybody, and thank you for inviting me to give this talk about basic and clinical research. My understanding this audience is going to be students and residents uh, entering into this medical career, and I hope to give you a broad overview and an outline of this vast subject area. I'm going to be briefly touching on a lot of topics today, so don't be worried if you don't understand the whole intricacies of research. I'll be putting in references in each slide for you to follow and read through. And there are also some additional reading material which you can potentially look at if you're interested to learn more. And I'm always available. Please feel free to email me at this address. This is my personal email address, and I'll be happy to respond to you. My talk is going to be organized in multiple topics. I'm going to be first talking about basic research and the importance of basic research in our evidence and knowledge base, followed by briefly on preclinical research. The bulk of our time is going to be spent on talking about clinical research because that is where our bulk of the evidence comes from our practice of medicine. I'm also going to touch about phases of a clinical trial for a new drug or device. Very briefly, we can talk about translational research and repurposing existing drugs for new indications as a special case of translational research. The foundation of evidence-based medicine rests on multiple layers. The first layer is your basic science, that is the broad knowledge base of understanding of our biology, followed by preclinical research in vivo models or animals, followed by clinical research, which is the broad information and evidence that we have on humans. This eventually, once we get interventions that help either maintain health or treat disease, gets translated into clinical practice. And the hope is that from clinical practice, we implement in a wide population to improve the health of a large population of patients. So what is basic research? Basic research is an investigation focused on basic understanding the phenomena or a natural process. So this is the goal of this research is to increase our scientific knowledge base. Sometimes it may be just curiosity to identify and describe a process that has never been seen before or understood before, improve our understanding of a fundamental process, a cellular or a genetic process. Sometimes initially there may be no obvious application in human disease, but like all information and knowledge, as we build upon it, it gives us a overall understanding of the underlying phenomena, and which in turn may actually end up giving us a potential application in human disease much far into the future. So for example, a fundamentally important process called RNA interference, this was first identified in 1998 in an animal or nematode model system called C. elegans. And I'm going to talk about RNAi or RNA interference a little later. But suffice it to say, from since 1998 to currently, we have come a long way in understanding this process and actually utilizing this in treating human diseases. Preclinical research is once we find a basic science finding or understanding of a phenomena and we have a potential clinical application, then we find more information by experimenting in cell lines. The first experiment we do in cell lines is called in vitro, and then this is followed by, once we have enough information from cell lines, we use that information to go to the next step, or in vivo, in model organisms. There used to be organisms like worms, like nematodes, Drosophila in mice, rat, and then primates. like in rhesus monkeys. And then there are other more sophisticated experiments in preclinical research that can now be done ex vivo. Ex vivo is a name given for human tissues, for example, on organ cultures like brain slices. 3D cultures is another interesting method. In traditional in vitro experiments, we do it on a petri dish. In 3D cultures, we do it in three-dimensional growth, which actually is more physiological. And then more recently, we have organoids. Organoids are basically miniature organs, or so to speak, that are derived from embryonic stem cells or induced pluripotent stem cells, or iPSCs. And these organoids are interesting in that they have multiple cell types that are all present in a full organ, and so we can do a lot of additional experiments that may not be really possible to be done in vitro or in a cell line, and which can also potentially not be done in ex vivo. We switch gears and talk about clinical research. So once we have information from the above preclinical research, the next question is to see how we can use this information to either preserve health or improve outcome from a disease. The Belmont definition for research is any activity that is designed to test a hypothesis and permit conclusions to be drawn, and therefore develop and contribute to a generalizable knowledge. So this could be theories, principles, or statements of relationships. It is an interesting report to read, and that will give you a little bit more information and nuance into what is defined as research. And this is important that you read the Belmont report if you're interested in doing any kind of clinical research. However, the regulatory definitions for research may be slightly different. For example, the COFEPRIS or the FDA or ANVISA in Brazil have slightly different relationship or definitions which must be followed if you're actually performing a clinical trial, depending on which country you're going to be doing this. The most important framework of clinical research is that it is framed in an ethical, there is an intact ethical framework, data integrity is preserved. In clinical research, once we have cleared the ethical framework and preserved data, can be either an analytical or a descriptive process. So in clinical research, for example, there's two different main classes. One is descriptive and analytical. And descriptive, let's talk about descriptive research. One of the smallest unit of descriptive research is a case report, where you actually describe a particular patient, either with a rare disease or a rare presentation of a common disease. And the idea of publishing a case report is to inform other clinicians of this interesting either a clinical scenario or a rare presentation of a common disease or a rare disease. So obviously, since this is for a single patient, the findings and results are not generalizable. This is simply to improve the knowledge base and inform your peers. The second class of descriptive studies, next step will be case series, where a case series is a set of patients that are similar to each other, and so there is a little bit more information now. Again, this is not generalizable because you're picking these patients based on a particular finding. And for example, early on, when HIV became an epidemic, we didn't know about HIV virus or AIDS. What was one of the first case series was Kaposi sarcoma showing up in these young patients in New York. Kaposi sarcoma prior to that was very rare, first of all, and not seen in young patients but much older people. So the fact that this is coming up in young patients within a short time frame suggested something else was going on, and that was one of the first signals that there was a new virus that was causing AIDS. Then the next step in description is basically like things like surveys, where you send out a population level survey and analyze the statistics on distributions of various issues. Population studies, for example, on outcomes of COVID-19 in Wuhan early 2020, looking at the number of patients getting admitted in the hospital, the number of patients getting admitted in the ICU, the age distribution, and things like that. It's important to understand that descriptive research is to help you analyze a trend. It may be useful for healthcare planning and to improve your hypothesis generation. So this may give you a signal as to where to look for, and so the next step after descriptive research is to do a little bit more analytical research, look at causation. Descriptive research does not cannot infer causation because you're not comparing it with anything else. You just have a population that you're describing. The answers the question of what are we, and not the question of why. Just to briefly recap, you have clinical research can be broadly divided into descriptive and analytical. And descriptive studies like case series, case reports, surveys help you to generate hypotheses, and following which you take those hypotheses and analyze in either form of an observational study. Observational, by the name suggests, you're observing and you're not doing anything to the population of the patient, so you're simply seeing what's happening. However, in interventional studies, you're actually intervening either by changing the environment or giving a drug or a device and looking at outcomes from there. So obviously, the risk for each kinds of studies are different. There's absolutely no risk for the patients in the either descriptive or in case series or surveys. And also, there is no risk or ethical issues with observational studies. Although, when you come to interventional studies, because you're doing something, there's a high element of risk to the patient. So you have to be very careful and make sure patient safety is always at the forefront. So let's talk a little bit more about analytical research. Like we talked before, analytical research can be classified into two different, one is observational and interventional. In observational studies, you do not control, the investigator cannot control the assignment of a subject into one or other group. The primary objective is to discover and quantify an association between exposure and an outcome of interest. So this can actually allow you a causal inference. However, observational studies can have any kind of multiple designs. It can be retrospective, meaning looking back in time, a prospective design, meaning look forward in time, or a cross-sectional design, meaning you look at one slice of time but look at a whole population or a subpopulation. On the contrary, in an observational study, you're actually applying an intervention. It could be a drug or a device or a modification of an environment, or like example exercise, and then you measure outcomes of interest. It's important to understand that interventional studies can be either randomized or non-randomized, but the majority of the evidence that we generate for our evidence-based medicine comes from randomized trials, and I'm really not going to be talking much about non-randomized trials at this point. Randomization reduces confounding and therefore reduces effects of unintended variables or unmeasured variables, and blinding reduces placebo and reporting bias. We'll talk about observational studies. There are three main groups or classes of observational studies. The first kind is a cross-sectional study, a cohort study or a case control study. In cross-sectional study, measurements done on a whole population at a single time point. The investigator measures the outcome and exposure of the study subject at a single point in time, and then can assess the association between the outcome and exposure. In the right here, as you can see, there's a target population from which a random sample is drawn, and then from that particular population you gather data and look at an exposure and an outcome, whether it's present, exposed and no outcome, unexposed and outcome, and unexposed and no outcome. So what a cross-sectional study will give you is a prevalence of a condition or a disease at a single time point. It does not give you incidence. In the example here, you can look at smoking and COPD diagnosis, where, for example, you take a population of patients and you look at the number of patients who have COPD diagnosis and the number of them who smoke, and using that you look at if there's a correlation between the two, or what is the prevalence of smoking in patients with COPD. There are several advantages of a cross-sectional study. Number one, and the most important, is that it's very quick to do and it's inexpensive, no ethical difficulties, and data on all the variables are collected at a single point in time. Multiple outcomes and exposures can be studied, and the most important part of a cross-sectional study is that it's easy to generate hypotheses that can be tested further, especially to create an additional in-depth research study later on. So this is a starting point. In some words, almost like a descriptive study, because on all descriptive studies, for example, have a cross-sectional design. I don't want to confuse you too much on that because they're two different. Cross-sectional, the intent is not description, but just to give you an overview of the similarity between the two kinds of studies. The disadvantage in a cross-sectional study is that you cannot measure incidence or make a causal inference because of the presence of what is called confounders. Confounders are variables that may exist, which seem to create an association between your variable of interest and an outcome, when in reality there may not be an association because this is something that you cannot measure. And so the associations that you may identify in a cross-sectional studies can be difficult to interpret, and you need to be very careful not to overcall associations and causality. Another important information that you cannot obtain in a cross-sectional study is that you cannot really investigate a temporal relationship between outcome and risk factors, because you're only following patients at one single point in time. This is not good study for studying rare diseases, because remember, you have to have a good prevalence in the population for you to actually get data from a cross-sectional study. Importantly, susceptible to biases such as non-response bias and recall bias. We'll switch gears and talk about the second kind of observational study, or called a cohort study. In cohort study, the design is basically you first select a population, which we call as a cohort, and then you follow them through time. So the cohort can be followed either prospectively, meaning from the present going into the future, or it could be retrospective, looking into the past. For example, where we have here in a cohort study is you look at exposed and unexposed, and then you look at the presence of disease or absence of disease, and similarly in the unexposed, the presence of disease and absence of disease. One example of a very powerful cohort study is the Framingham Heart Study. This began in the early 1948 with more than 5,000 patients, or I should say subjects, from the Framingham, which is a town in Massachusetts in the East Coast. And this is a still ongoing study with over 3 generations of cohorts. This study alone has generated more than 3,000 papers. The important findings that the study gave us is, for example, is the smoking and cardiovascular risk as a big cardiovascular risk back in the 1960s, and then hypertension and the association with cardiovascular risk in 1970s. Cohort studies, because they're looking at exposed and unexposed, you can actually give you a relative risk. The relative risk is a number where you can, it's a risk in exposed versus risk in unexposed. So by comparing the risk in exposed and the risk in unexposed, you give a relative you get a relative risk. So, and here is your URL if you want to look a little bit more into the Framingham Heart Study. This is actually a very interesting read if you'd like to look at it. The advantages of a cohort study is that you can investigate multiple outcomes and maybe associated with multiple exposures. Because remember, you're looking at a population and you're going to see them through time. You're able to study the change in exposure and outcome also over time. It's very good for examining rare exposures because you're going to be following those patients, the exposed patients. You can measure the incidence of outcome, which is important, and you may be able to infer causality, but this cannot be said with certainty, but you can potentially offer some insight into what could be causing these issues. The disadvantage of a cohort study, obviously, is just they're super expensive and time intensive. You need measure all variables, susceptible to loss of follow-up compared to cross-sectional studies, because the patients have to have a very good relationship with the study team to keep coming back over and over and over again over a long span in time. Patients may move out of that area, so there are a lot of attrition that could potentially happen, so you need to have a large n number. And retrospective cohort studies, for example, are easy to do, but you have less control on the variables and are subject to what we call as recall bias or informational bias. Because you're going to, I'm just going to briefly talk about informational bias here. Remember, retrospective studies are you looking back into the past on data that you already have. So when you have a retrospective cohort, the initial information that was procured may not have been for the purpose of your study. So there may be some missing information or variables that you may not be able to access at this point in time because we don't have access to them anymore. So there is that's called when you have a lack of information, that's called an information bias that may affect your outcomes of your study. The other issue is the cohort studies and the disadvantages. That's confounding variables are a big problem, and unlessing data in a cohort compared with the randomized control trial. Next, we'll talk about a case control study where you have a case or a patient with an outcome, a controls without an outcome, and so on. Then you go back and see the presence or absence of an exposure, and compare that with the presence or absence of an exposure in a control. So based on the design of this study, it's actually eminently suitable for rare disease or disease with a long latency period between exposure and disease manifestation, because you're already beginning with a case or a patient with a disease. The another advantage is that it's possible to study several exposures, and particularly suitable and exposure data is hard to obtain or expensive. Cost effective because it works with a small sample size and can be performed in a short period of time, and no follow-up is needed, and therefore particularly suitable for a dynamic population, unlike for a cohort study. The outcome measure that you're looking at is an odds ratio, where you look at odds of exposure in cases and the odds of exposure in controls. And this odds ratio will give you an idea of how much of your exposure is contributing to your case. For example, lung cancer patients and smoking, where you look at outcome as you look at patients with lung cancer and without lung cancer, and then you look at presence of exposure between the two groups. In which case, in this particular case, the presence of smoking. The advantages of a case control study is that it's suitable for rare disease or long latency with exposure and disease manifestations. So we talked about before. It's cost effective and really no need to follow up because it's a single time point. The disadvantage of this is that it's not suitable when exposure is rare. Exposure status is difficult to sometimes ascertain due to recall bias. Incidence and risk cannot be directly computed. More importantly, the causal relationship between exposure and outcome cannot be determined because of a potential bias issue for control group selection. And you can look at the reference below for full details. They actually have an excellent write-up on that, on the intricacies of on the different kinds of case control studies, which we won't get into this today. And for further reading, I really recommend that you look at this collection of articles in July 2020 volume from CHEST. I think this is available for free, and it has an excellent overview of statistical considerations and study designs of different kinds for a clinical study. This is also a second further reading of this book. This is an excellent textbook on designing clinical research by Browner and Newman. This is published by Walter Provos in the 5th edition. I would highly recommend that you look at both of these resources if you're planning to do any kind of a clinical research or designing clinical research. The next kind of research that we talked about from observational is called interventional studies. By interventional studies, that you intervene or you do something to the patient or the population, and you're looking at outcomes. So the classic interventional study is a randomized controlled trial on a RCT, which you normally call as a clinical trial, in which you have a single intervention and you observe outcomes. The main reason why we love randomized controlled trials for our evidence base in medicine is that randomization as a process reduces confounding variables. This is the only way to accurately say for sure that can remove confounding, because by definition confounding variables are something that you may not be aware of. So by randomly allocating patients between the groups, you're also randomly allocating the confounding variables, and thereby eliminating the confounder. So the second portion of a randomized control trial is control. Control is a population that does not get the intervention or the treatment, and therefore you're going to see whether it allows us to infer a causality of treatment, meaning whether your drug or the interventional choice is making a difference to the outcome. A third portion of RCT is blinding. Blinding is just a fancy way of saying when the patient and the investigator do not know who is getting the treatment and who is getting the intervention. This is very important because there is a condition or an effect of placebo effect where patients feel better when they know that they are getting treatment, and it's the only way to remove the placebo effect is to blind patients. And also blinding the investigator is important because it eliminates bias. So by doing all these three, by doing a randomized control double-blind trial, will give you the gold standard evidence of if a treatment or intervention is helping or not. And sometimes RCTs are the final say in, in EBM or evidence-based medicine, and without this you really cannot proceed to take a treatment and apply to a population. There are other interventional study designs instead of the classic one intervention, one outcome. For example, a little bit more complicated study design is a factorial design, where you can address more than one question in a single study. Example, you have a drug A and a drug B, and then you look at the risk of, for example, cancer and diabetes, and the effect of A and B on these two outcomes. So you can address more than one question in a single study, so somewhat cheaper to perform the study. However, the statistical analysis and the other details of the study protocol becomes a little bit more complex. An active control trial is an example, or in other words, is an equivalence or a non-inferiority trial. For example, say you already have a medication that is working, as a statin used for cardiovascular risk reduction in patients, and you have a new drug that you want to bring to the market, a new statin drug, and you want to look at how good is your drug compared to the old drug. So we know that if you don't treat these medications for say 2 or 3 years, and looking at the outcomes, it is ethically not possible or feasible to do such a trial when you already know there's an existing treatment that works. Therefore, the only way to proceed further would be to compare with the standard of care and see that your drug of choice or the new drug is at least same as the old standard of care. What you hope to find is that your new drug is superior, and then you have a better drug to go forward with. An adaptive design is a different kind of a design where instead of having a fixed protocol for the fixed treatment, and then if you want to try 4 different drugs, by classical double-blind RCT, you have to run 4 trials. However, in adaptive design, you build into the protocol a method in which you can change based on interim analysis. So, for example, the COVID platform trials are an excellent example for an adaptive design, where they assess multiple treatments. So first they try treatment with a drug A, and you see, compared with standard of care, if the A is better. And then if A is better, then you use that as an active control, and then check drug B. And in that way you can do you can assess multiple new treatments or interventions, and then find the best possible treatment. A crossover design is an interesting design paradigm where each patient can serve as their own control. So for example, a patient takes a medication or a drug for 10 days, and then you give a washout period for a week, and then you switch them to a placebo group, and then they'll do this and switch back. So that way you're looking at all the treatments, all the patients will get a treatment. Obviously, for this to work, you cannot have an outcome, a short-term outcome. N of 1 is a special part of or a special case where you have in a trial a single patient, um, and you treat them as a single patient clinical trial. This is a little bit more complicated to get into, but there are interesting, uh, if you want to read up, it's actually a very interesting paradigm to look at a framework where you can use or you can glean useful information from actually treating a single patient in a trial, in a blinded trial. Then more recently, we have Bayesian clinical trials, which are kind of an adaptive design, but in which the statistics allow you to actually not have a fixed n number, and it's much more cost effective to perform. So we'll switch gears a little bit and look at the core concepts for designing clinical trial. The first and most important is a correct research question, is what is the purpose of the study and how are we going to study it. So it's important that you write down the question in a concise single sentence. And the second important step on designing a clinical trial is to look at the background and significance. What is it that you are attempting to address? What is the prior knowledge in this area? And so this will involve a lot of legwork. You have to look at papers that has come before, see what has been done, and to try to build upon that. And then once you figure that out, you need to try to figure out what is the kind of study design that you're going to be using to address this clinical question. Is it going to be observational? Is it interventional? What is the study population? What are the potential challenges? What are the inclusion and exclusion criteria? This is extremely important because this is kind of the inclusion criteria will tell you what kind of patients you're going to be selecting for the study. It's all, and the exclusion criteria is even more important because what kind of patients you do not want in the study, because when you are having an intervention, if you include everybody in the study, you may have too much noise. At the same time, if you exclude a lot of potential participants, you may not have enough n number. So at some point you have to have a balance between which patients you can include and which patients you can exclude. Exclusion could also be important, that you have safety exclusions. For example, if you have a drug that can have toxicity in patients with kidney failure, you want to exclude them. So those should be automatic safety exclusions, but they should also be robust to have a relatively homogeneous population to address the question that you're answering. The next thing you need to look at is measurement, and how are we going to measure the clinical variables, how are we going to account for confounders, and how are we measuring outcomes. Statistical considerations and generalizability. It's important to see that the study results are actually applicable to a larger, more general population. So you need to make sure that your study population is at least similar to the population that you're going to be targeting eventually. For example, if the studies are only done in one particular race, they may not be always be obviously and easily generalizable for a more diverse population. And this is sometimes an issue. For example, the Framingham Heart Study mostly only looks at whites, and so some of those study results, although generalizable, there is some concern about generalizing to a more international population. Once you have the answers to all the above, then you need to write the study protocol, and that's once you write in black and white, and that's kind of it's set in stone, and that study protocol will now help you to get funding. Without funding, obviously you cannot do any studies, so it's important that you do all the legwork prior to getting your funding. Important point which I have discussed before in any clinical trial is consent. The patient or the participant in the clinical trial should be completely informed about potential risk and benefits of the trial or participating in the trial. There should be no coercion, either obvious or unintentional. You need to make sure that it's completely voluntary. Participation should be voluntary for the in a clinical trial. Safety is of utmost concern, and efficacy, outcomes, and data analysis plan must be clearly determined prior to initiating the trial, as put in your study protocol. Regulatory requirements must be followed. If you're doing a new drug, you need to have an investigational new drug application to the regulatory body. You have to have ethical approval from the IRB, and the regulatory body should be notified and accept your study design, because if you do a study and submit the data which finally the FDA or the COFEPRIS says it's not going to be useful, now you wasted a lot of time and money and effort. So involve the regulatory body early on, especially if it's going to be a large trial, for any advice in the outcome of the studies. Data integrity and security has to be is of utmost important, and privacy of the participants has to be respected. The whole research team, anybody who handles research information, so that includes not just the principal investigator, the research coordinator, nurses, anybody involved in research, the students, the residents, all have to be trained in GCP practices before initiating a study. There is a set of points that is called the good clinical practice, which is application of ethics and integrity to human trials, and this is based on the ICH technical requirements for new drugs. So you can go to this website in ichgcp.net. Basically, it gives you a set of standards for design, conduct, performance monitoring, and basically how you conduct a trial and what needs to be in place to make sure that ethical there is protection for the rights, safety, and well-being of the subjects, and make sure that the data is of credible value to society. I'm not going to go into the details, but the few principles of ICH GCP is that the trial should be conducted in accordance to ethical principles that have the origin in the Declaration of Helsinki and consistent with GCP and applicable regulatory requirements. Before a trial is initiated, any foreseeable risk and inconveniences should be weighed against an anticipated benefit to the individual or the society, and the trial should only be initiated if the benefits justify the risk. Safety of the well-being of trial subjects are paramount, and there's so on and so forth. There's a lot more information that you can look at, but these are very basic principles that must be adhered to perform a valid clinical study. I'm going to switch gears now. Go briefly talk about phases of a clinical trial for a new drug or device. Now I'm going to be switching gears and we'll talk about the phases of an interventional or a clinical trial. Say for example, I have a new drug that I want to bring to the market. There are several phases that we have to get through before it gets to the market and goes through the regulatory authorities. The first phase is called phase 1, where we get safety data. So where you have get information on safety, tolerability, finding the dose, and also pharmacokinetics and dynamics in humans. So this is called the first in human studies. Usually the n numbers are small, less than 50. Phase 2 is looking for efficacy and safety, where you have data on some efficacy, usually less than 100. In this particular case, in phase 2 series, you don't really have any placebos, so you don't really look for a comparison, but you're looking at some outcome for efficacy. You look at adverse effects and continue to gather more safety data. Sometimes phase 2 can be divided into a phase 2A, 2B. 2A is for dosing with multiple dose ranges, and phase 2B is for efficacy, or called proof of concept studies. Once you have a proof of concept of phase 2, the n numbers are usually a few hundreds. Go to the phase 3. That is the final step where you look at therapeutic efficacy. And so here, this is your standard, gold standard, double-blind RCT, where you look at the standard of care compared to a standard of care or a placebo. The n numbers are usually a few thousand, hundreds of thousands. If your drug works in phase 3, then you mark it and then you look at phase 4. Phase 4 is post-marketing studies. This will continue to collect more safety data, real-life data for efficacy, and also rare adverse effects. Translational medicine is basically a name given to the process that we have been doing all along, from basic to preclinical to clinical research to clinical practice. But this process actually takes a long time. So if you look at the time course, for example, for a new drug, when you first identify a target in the lab, it takes around a year and a half. Then you look at more than 10,000 candidates that you screen, of which maybe a few, around 250, gets comes to the called lead optimization stage. Then you take it to a preclinical test in model organisms, maybe another 10 to 20 candidates, and then go to phase 1, phase 2, phase 3. This whole process can take more than 15 to 18 years. By the time you finally launch, you have screened started from 10,000 candidates and maybe gotten to 1. So this is a long process, and the cost and the effort involved is tremendous to get one particular market drug to the market. In translational medicine, as we talked before, you start a basic science or a research basic research finding, and you try to translate it to human early on, but it's an iterative process. So you take it, so this is a team effort where you just not one a clinician or a researcher is doing this. There's a team of basic scientists, pharmacists, clinicians, clinical trialists, all of whom work together, and we try to compress the amount of time from 15 years to hopefully much lower by having a pipeline in place. And you can go back and forth seamlessly from a preclinical study to a phase 1, back to the preclinical study, and optimize lead optimization, go to a phase 1, phase 2, and phase 3, and so on. So this is called the value, and this particular thing, translation from basic science to human studies, is very risky and very inefficient based on our current policies and processes. So this needs to be optimized, and this whole process of optimizing this mechanism from growing from basic science to the clinic is called translational medicine. It's a team effort, and one example for translational medicine I will talk about is RNA interference. This was first identified in C. elegans. C. elegans is a worm or a nematode back in 1998 by Craig Mallow and Andrew Fire, where they found that when they inject double-stranded double-stranded RNA, it caused post-transcriptional gene silencing. This opened up a new field of study in basic science called microRNA, small interfering RNA, and also the field of RNA biology and RNA editing. Soon after, in 1998 and 2001, they found that this process was conserved in mammalian cells. They proceeded to show it in a preclinical mouse model and adult mice that is conserved. And in 2006, there were clinical trials that were started for potential drugs. This is a new class of drug because it doesn't it is not a particular drug itself, but a process. And this particular process can target any gene and reduce its levels. So obviously it has tremendous potential. Finally, in 2018, almost 20 years, the first RNA-based therapeutic patisiran was approved. This is for treating TTR amyloidosis, where this RNAi drug can reduce the level of TTR or transthyretin in the liver, thereby alleviating amyloidosis in this condition. Later in 2019, givosiran was approved for acute hepatic porphyria, and then 2020 lumisiran and inclisiran, which is a PCSK9 inhibitor for hypercholesterolemia and familial hypercholesterolemia. So this process is, as you can see, a long process involving hundreds and thousands of researchers and a lot of time. Done right, it can potentially improve human health. And a special case of translational medicine is called drug repurposing, where we already use an approved drug for a new indication. So this is remember the biggest time that takes from step 1 to coming to a phase 3 trial is all the intermediate steps. But for an already approved drug, you can already bypass all those steps, go directly into a phase 3 trial or a phase 2b, which is a proof of concept efficacy trial. So before we can do that, however, there needs to be several processes that you need to keep in mind before repurposing a medication to see whether the data they have in vitro support, whether the drug has an appropriate pharmacokinetics and dynamics for the condition that you're studying. Does the proposed dose or formulation, is it similar to the approved indication for the drug? Whether an unmet need, or is there any alternative options? So by bypassing the initial bottleneck for phase 3 trials, you can actually potentially get this drug to the patients who need them if it works very quickly in a clinical trial. The problem is repurposing. There is a lot of intricacies. For example, if not done right, you know the story of ivermectin for COVID, where we didn't really, there was a lot of hype. The in vitro data showed that the ivermectin was working, but what some people didn't realize, that the levels of ivermectin that you need is almost 50 to 80 times more than the clinical doses that we use, and that is a conservative estimate, and that is a toxic dose for humans. So unfortunately, after all the hype settled down, more recent RCTs have shown that ivermectin doesn't really help in treating COVID-19. So it has have to be careful repurposing drugs, but if done right, it can potentially save a lot of time and give a new indication, and potentially help patients very quickly and rapidly. And this is just showing the same thing, where by appropriately repurposing, you bypass all these initial bottlenecks, you cross the valley of death, and you can actually go maybe directly into a phase 2B or a phase 3 trial and translate into practice within a span of a year to 2 years. And thank you very much for letting me talk to you about clinical research. I know this is a very heavy topic with a lot of things that we touched upon today. Please feel free to reach out to me if you have any questions. Thanks.